Jason Smith and Erica Fulham. Vicky has kids at Countervisor, and Erica went to Countervisor. So we have a lot of Countervisor time today. So I'll turn it over to them.
I graduated a really long time ago, in 2002, so it's a lifetime ago.
So my kids go here, so I have Evan Smith, Dylan Smith at the high school, and then Casey Smith at the middle school.
And Vicki and I work together at GSK. I've been at GSK for 13 years, went there right after I graduated from college.
And I've been there 11 years.
Yeah.
And so off and on, worked in the industry for, actually, which one I do?
Something like that. And we only recently figured out that we actually had this link to Countervisor, so it's been kind of cool.
Yes.
So thanks for having us today. So Vicki and I both work in biopharmaceutical manufacturing at GSK, and I'll talk a little bit about what GSK makes and what biopharmaceuticals are as opposed to regular pharmaceuticals, and talk a little bit about the manufacturing process, because that's what we do in our jobs. And then we'll talk a little bit about career path into that, if that's something that's interesting to you. Okay, so stop me with any questions along the way if you have any. We'll get started.
I have to play it.
There we go. Thanks. All right, so what products do we make? So we make, there's a couple different divisions of GlaxoSmithKline or GSK. We make prescription products, consumer healthcare products, and vaccines. So I just put up a couple of examples, ones that you might have heard of. So Advair, if you've ever heard of that, Brio, Ventolin, a lot of respiratory products for people with asthma and COPD. And then Imitrex, which is a migraine medication. Consumer healthcare, if you use Aquafresh toothpaste, Theraflu, Nicorette, probably nobody here, but vaccines. There's a Shingrix vaccine, which is relatively new, that treats, that's vaccine for shingles. And then a couple of other ones, Rotarix, which is for babies to protect them against any stomach viruses, Cervarix for HPV. So lots of different products. I just threw a couple up here. It's a pretty big company. We work in the pharmaceutical division of that. And as I mentioned, we make biopharmaceuticals. I'll talk a little bit about what those are. So in the prescription pharmaceuticals section of the company, so any pharmaceutical product has what's called an active pharmaceutical ingredient, or an API. So that's any medicine that you take. And what that does is it enables the medicine to treat whatever the disease target is, so whatever you're taking the product for. Depending on the ingredient, it can either fall under the category of a small molecule or a large molecule. And I'll talk about what that means exactly. So small molecule drugs have kind of been like the traditional pathway and has long been the basis for drug development. Manufactured using chemical synthesis, so this is just a series of chemical reactions to get to the final product. And this is kind of the traditional quote-unquote white pill, not always a white pill, but pills that you take for medication would be a small molecule product. An example would be acetaminophen up here, which is Tylenol. So it's a relatively simple compound, and that's just a view of like what the molecule itself looks like, and then the pills themselves. Large molecule drugs, on the other hand, they're also called biologics or biopharms, so I'll refer to them a lot today as biopharms. And really what that is it's a protein that has a therapeutic effect on a specific disease target, and these are typically what are called monoclonal antibodies. Anyone ever heard of a monoclonal antibody? Or antibodies in general? Kind of, sort of. So produced from a living organism, so instead of it being just like a series of chemical reactions, it's actually grown up in live organisms and cells, or in the yeast or bacteria or animal cell line. And these are typically administered through an injection or an infusion, so it's not a pill that you take, it's a different route of entry into your body. And they typically target autoimmune diseases and cancers. So some autoimmune diseases might be like rheumatoid arthritis, lupus is another autoimmune disease, and cancers. And so this is an example of what a monoclonal antibody looks like. And I'll talk a little bit more about what that is. And then just an example of a biopharmaceutical drug, which I mentioned is usually an injection or an infusion. Good so far, any questions? Okay, so what are the benefits of the biopharm product? So this is a relatively new field, probably has been a little bit more booming in the last decade, I would say, and continues to be an important path for pharmaceutical companies and for healthcare. So the benefits of a biopharm product is that they tend to have a higher efficacy, which just means how effective it is in the body at treating the disease. And the reason for that is it's more directly targeted to it. And when you take like a pill, it usually kind of goes through the whole body, but if you actually take a biopharm, it will target that specific disease. So what that means is you get less undesirable effects or side effects, and typically a lower dosage is required because it's able to just target the disease area. So some of the challenges with it is it's really expensive to manufacture, and it's because of the complexity of the manufacturing process. And it basically means then, higher drug prices, longer approval times with regulatory agencies. Since this is a newer kind of field for pharmaceuticals, regulatory agencies are still learning about it themselves. So for the FDA or in other countries, their equivalent to the FDA, it might take a little bit longer for them to approve it because they're still learning about it themselves. Success in the past decade, specifically in the autoimmune disease space, which I mentioned, rheumatoid arthritis, also in respiratory. We've seen several biopharm products come through, and you're starting to see more and more of them targeting cancers, which is really exciting. Pharma companies continue to invest a lot of money into the development of these kinds of products because they really do see this as the kind of future of healthcare and where we're moving towards in terms of medicine. So it's a really exciting place for Vicki and I to be working in to see that kind of growing field. The next wave of technology, I talked about monoclonal antibodies, so MAB is what that means, MAB. We're starting to see now that they're actually joining together monoclonal antibodies with a small molecule target to make it even more effective. And that's kind of an even newer field that you're seeing actually more effective drugs that can target and kill cancer cells more readily. So that's an exciting space to be in. And we're lucky enough to work on a product that's moving in that direction, which is new for us. So it's very exciting. Any questions on any of that? Yeah.
If you see like a drug name with MAB at the end of it, is that, that's
Yeah.
You're ahead of the game. That's good. And yeah, that's absolutely right. So whenever you see anything on TV or anything that's advertised that has that MAB at the end, that's what that means. It's a monoclonal antibody. Good catch.
And maybe just another way to think of it. So when you think of a small molecule, a chemical, so when it comes into your body, it's a chemical that goes in, so it might not target different areas. So you might have side effects because it might impact other parts that are healthy, and it might cause other side effects that you weren't thinking of. But with the monoclonal antibodies, because you create antibodies, and we're making it a protein, which is something that's naturally produced by a cell or by a yeast or something else that's living, it can target. So antibodies are very target specific, so it can go after something very specific that we want it to go to. That's why they're more effective, and that's why you don't see as many side effects with them.
So this is the slide, yep. So you can see all the MAB products. And I just threw a couple up that maybe you might have heard of or seen commercials for. I know Keytruda is a relatively new one. I see commercials for that all the time. And just some of the indications. So you'll see a few for RA, rheumatoid arthritis. There's a couple of GSK products, Benlysta, which treats lupus. It was actually the very first lupus treatment in over 50 years that wasn't a steroid, which steroids, if you've ever known anything about steroid treatment, they're very, very tough on the body, a lot of harmful side effects, and just a really tough way to kind of live. This is the first product that kind of came onto the market to actually treat lupus. So for those patients, it's been life-changing. And then our other one that we manufacture is Nucala, and this is for severe asthma. And then a couple other ones that you might have heard of, Xolair is another one, also for severe asthma. So yeah, anytime you see that, and I'm seeing more and more of them actually. My mom recently, she suffers from chronic migraines. So pretty much every day, most days out of a given month, she has a migraine that keeps her from working or just functioning normally. And she just kind of went to her doctor and he put her on this new product and she was telling me about it and I was like, well, what is it called? And she told me and it was actually a biopharm product, so I got really excited because it was the first time I actually knew someone that was taking one. She didn't really understand why I was so excited about it, but I was like explaining to her, why it's better for her because it's more targeted and it's been really helpful for her. So it's just great to see it sort of, coming into people's lives more. So what is a monoclonal antibody? So I was going to give just a quick, I don't know if I can click on the link from this thing, but if the sound will work, but we can try it. If it doesn't, we can just kind of talk through it. It's a 1-minute video, so it's not long, but just to kind of give you an idea of what, available. Sorry. But a few of you nodded heads when we talked about antibodies. Anyone learned about them in school yet? Yeah? Anyone care to be brave and say what they can do?
So they're kind of like these little things that some white blood cells shoot out at like disease cells to show that they're disease cells, and then the other white blood cells target them.
That's exactly right. Good job. That's great. Yeah, and the way that they kind of look, and you'll see this in the video, those little Y-shaped things on the ends of those little prongs on the antibody, very specific markers, and they go to their very specific antigen, and it's almost like a puzzle piece fitting together. They're very, very specific for that disease. And so they'll go and actually find, so the B cells, which are white blood cells, will go and actually produce those antibodies and go find the disease target and clear the disease if it's working the right way.
Antibodies are proteins that play an integral role in the complex system of specific immunity. They bind to and inactivate foreign proteins called antigens. Each type of antibody binds to one kind of antigen molecule. B cells are one of the 2 types of white blood cells that make antibodies. When a bacterial cell enters the bloodstream, proteins on its surface act as antigens and trigger response by the immune system. These antigens will bind to the unique B cells that have the corresponding antibodies on their surfaces. These B cells now divide. Some continue to divide, producing many plasma cells. Plasma cells secrete large quantities of antibodies into the bloodstream. This division of only the appropriate B cells is called clonal selection.
Thank you. So you're spot on with your definition. Thanks again. So it makes sense that it's kind of, like I said, a very specific, antibody for a specific antigen. So when we talk about biopharmaceutical products, there are folks that work in research and development at our company and all these other companies that will work through genetic engineering to create and discover antibodies that will target different diseases, to put it kind of simply. So an antibody, again, is a protein that's produced by the immune system that attaches to and counters a specific antigen. A monoclonal means that it's all clones of a unique parent cell. So the original one that kind of exists, it clones itself, so they all are exactly identical. And that's important when you're taking, if you think about medication, you want it always to be the same, so it's always doing the same thing. So biopharms, or mAbs, they still target a disease process, as Vicki said, like the small molecules do, but their mechanism of action is a bit more complex. Depending on what the medicine is, what the mAb is doing, it might trigger certain reactions in the body, or it might just disrupt the disease process. It kind of depends on what it's treating. And then just to give a little bit of a comparison, so this is the molecular makeup of aspirin, molecular mass, I should say, of aspirin. And then when you look over here at an antibody, it's much bigger, it's much more complex, there's a lot more to it. So when it comes to the manufacturing process and making sure that the drug is the right drug and testing for it, everything kind of gets a little bit more complex with that. And then this is just kind of the overview of that little Y-shaped antibody. And so this is kind of up top here, it's called the Fab region, and that's the one that binds to the antigens, that has the really specific puzzle piece that I talked about that will attach to its antigen. And then the bottom is called an FC region, which is the more kind of common among the mAbs. Anything to add, Vicki? Any questions on that? So our manufacturing process, so what Vicki and I do every day. So I mentioned before that this is done through live cells, so it's called cell culture. Have any of you had any experience doing any kind of cell culture in the labs? Yeah? What kind of cells do you remember? No? Doing it in like maybe flasks and media? Familiar with that? So basically it all starts with a mAb cell line, or we call it a cell bank, that's been genetically engineered to secrete your medicinal protein, whatever medicine it is you're making, whichever mAb. They're cryogenically frozen in liquid nitrogen to preserve that cell line. And I mentioned before, it can be bacterial, it can be yeast, or it can be mammalian. So a lot of times people work with what are called CHO cells, which are Chinese hamster ovary cells, or HeLa. So if you've ever read anything about Henrietta Lacks, anyone read that book? Yep. Yeah, a really interesting story behind that, but HeLa cells are another big one that's used in the industry. So basically, when you start your manufacturing process, you thaw the cells from your cell bank and you place them in shake flasks with media. Media is basically food for the cells to keep them happy and growing, and that's called inoculation. So for those of you that have done that, it's something you already probably know. And then you place them in an incubator, and they're called shake flasks because they sit on a shaker table to keep the mixture moving, to keep the cells happy. So warm and fed cells equal happy cells that will divide and grow, which is what you want, because you want them to grow up so that you can make your medicine. And then we scale it up. So we start in these little shake flasks from the tiny vial in the beginning, and you work your way up through what are called bioreactors. And basically they're just bigger versions of a shake flask that have more mechanical elements to them. They're usually in big stainless steel reactors that can get very large, up to 20,000 liters in some cases. Some of the scales that we work with in our company can go up to that high. In our manufacturing facility, we go up to 5,000 liters. And we have a sister site in Rockville that goes up to 20,000 liters. And basically what happens is the bioreactors within them have what are called agitators, so that's what these things are kind of in the middle here. And it's basically just a big mixer, and it stirs the cell culture to keep the cells suspended. So they're not clumping together, they're happier when they're suspended and freely moving, as they would be in the body. We monitor very closely a few things, the pH, the temperature, their oxygen levels, carbon dioxide levels, to make sure that everything is at the right level for the cell and for the protein. And this gets very specific. They're very tight ranges that they have to be controlled within, and that's all part of what our R&D, research and development folks kind of determine before it comes to us to manufacture it.
It's not just to keep them happy, but it's also not to kill them too, because they are living things.
Yeah, exactly. Very good point. And then each day, it's not like you just kind of let it run in there. Every day a sample is taken, and we actually look at the cells under a microscope and count them to make sure that they're at the right cell count level, to make sure that they're still happy, that they're not dying. And then we make adjustments as needed. We also do it to make sure that, as they kind of grow up and get bigger, we know when to go to the next bioreactor. And this process probably takes, from the time you thaw the cell until you get to what's called your production reactor, your biggest one, is typically about a month and a half, I would say. So it's a kind of a slow process. It's not like a chemical reaction like I mentioned with the small molecules, where you can kind of do that in the lab and kind of go at the pace you like. Unfortunately, you can't rush biology. The cells are just going to grow as fast as they grow, so you have to sort of wait for them to admit. So it takes a bit of time. And then once the correct volume is met, so like I mentioned at your production reactor, it's time to then harvest your cell culture. And so it's harvested from the bioreactor, which basically means it's collected, so it's taken out of the bioreactor, and then it goes through what's called centrifugation. Anyone ever used a centrifuge in the lab? Yeah. A couple people. Anyone know what it does?
It separates the cells.
Yeah, it does. So you basically have this big mixture with the cells kind of floating around in the media. So what it does is it'll actually, and this is just kind of a simple graphic to show that stuff separates out your cell debris, so all the stuff that you don't want, because remember your protein that you're making your mAb, it's actually secreted from the cells. So you want to just get that mAb, you don't really want all the rest of the other stuff, so you do centrifugation so you can kind of pull down by weight. So basically centrifugation means it spins really, really fast. And so it pulls all the heavier weight cell debris out of the mixture, and so you just have your mAb in the mixture that you want. And then it's filtered to kind of clean it up and make sure that you're getting out any bacteria, viruses, things like that. And then you move on to what's called purification, the next step. So protein purification is achieved by performing a number of column chromatography steps. Anyone done chromatography? I'm going to keep asking you these things, sorry. It's early, I know.
Julie, you have.
She
It's just
wanted
funny.
it. Yeah, so chromatography, there's a couple different versions of it, and I'll just give like a couple of examples, but it's a purification method where you get your desired product, the mAb in this case, is separated out from the rest of that harvest material. So you've done your centrifugation, but you're still not quite done yet, it's not purified yet. So basically it's impurity removal, so anything that's not the mAb you don't want to have in the medicine because it could be harmful to the patient. And that's governed by the FDA. There are a lot of rules around what we have to remove. Vicky's lab team does a lot of testing to make sure all those things happen and all these things are removed, right?
Yes. So with that, I was going to just add on to that. So when we're separating the product, when it's running through the chromatography, there's a number of ways to do that. You can adjust the pH so that your product flows through and the debris stays back. And it separates the product that way. So if you've ever seen chromatography or separation, anyone ever done a filter experiment where you add water and you look at the way that the ink drifts across there, you can separate the colors, like if you had a mixture. Anyone ever do anything like that?
Most of them have probably done it with like leaf payments.
Yeah.
Like when you do a photosynthetic.
Yeah. So just it's a matter of separating things, and you do it so that you can get your product at a certain time, and we have criteria that fits that. So things like the cell debris, so the cells actually will have different protein that you don't want there. It'll also have DNA that you don't want in there. All these things can cause reactions in the patient and can cause issues if people are allergic to, like if you use a yeast cell, if people are allergic to certain parts of that, then you want to make sure that you're separating that out so that you don't have any issues with it.
Yeah, thanks, Vicky. So at the end of it, you get pure isolated protein, which is what you want, so you don't have anything else in there. And usually with chromatography, you use specific properties of the product, as Vicky kind of alluded to interact with specific properties of the resin. The resin is like what's used to do the column chromatography. They're basically these really fine beads, almost looks like sand, and it's packed into this cylindrical column, and you kind of pour your mixture through there, and it actually acts as a filter, just to kind of visualize it a little bit. I'll give an example of that. So one way that we do it is using what's called affinity chromatography. And this is a little diagram, simple diagram of what that is. So I talked about the resin, so that kind of sand material. This one in particular is made of glass, really tiny glass beads. And on those is fused what's called an affinity ligand, which is basically an antibody itself that has an affinity to this region of your mAb. So if you remember in the beginning, I talked about the kind of structure of the mAb, and this long part of the Y is common among many mAb proteins. So this actually will have an affinity to that. So you're actually attaching an antibody to an antibody, which is kind of cool. And what that does is it basically in that mixture, it will find your protein and bind to it. So when you pass your mixture through, which has your antibody, the green parts here, and then all these other impurities that Vicky kind of talked about, and you pass it through, all of your mAbs that you want will bind to that resin and stay on the column, and the rest of your impurities will flow through and you can throw them away. And then you can actually debind it from the resin, or what's called eluting or elution, and you collect your purified mAb, and you do that by changing the pH, as you mentioned, Vicki, using buffers, which you guys have probably used buffers for things like pH testing and things like that. So very controlled pH that will kind of make the pH change, and then your protein can come up and you can collect it. Make sense? Any questions at all? Okay. So that's kind of, in a nutshell, how the process works. I mentioned before that it's really expensive. Some of the reasons for that you might be able to guess is that it takes a really, really long time. So that whole process, that upstream process where you're in cell culture is about 1.5 months, and then you're another week in that downstream purification process. And that's not really the end of it. Then it has to go and get filled into vials or syringes, whatever the medicine is, however it's administered. So it takes a lot of time, so that's a lot of people and time in the manufacturing facility. as well as the materials that we use. So the media that feeds the cells in the cell culture, as well as the resins, are very, very expensive. Oftentimes they're very specialized for your product. So the companies that make them, it's very expensive for them to make, so it's very expensive to buy. So you can kind of see why it creates a lot of cost. The testing then that Vicky's team does, oftentimes there's a lot of specific reagents, right, that you have to use that are really, really expensive too. So it just drives up the cost, and there's always, in working in the field that we do, a big push for us to find ways to reduce cost, because it's really not, you want people to be able to buy it and take the medication, so we're always looking for ways to actually bring down the cost so that the price can go down. But it's a big challenge across the industry. And then I was going to talk a little bit about good manufacturing practices, or GMPs. Anyone ever heard of that term before in the labs? Or GLPs, good laboratory practices. So this is something that's really, really important in any manufacturing industry. It's mandated by the government and the FDA to regulate manufacturing and product testing to ensure the product is effective and safe every batch, every single time. And that means a lot of different things. So it's minimizing or eliminating any instances of contamination, mix-ups, and errors. So think about the process we just talked about. There's a lot of potential for contaminating your cell culture or contaminating your purification process. And because you're actually injecting this into someone's body, it's extremely important that you are paying close attention and have strict controls in place. This protects the patient, of course, from a product that's not safe or worse, or not effective or worse, not safe. So key components of GMP, just kind of broke it down into 3. So documentation is a huge one. So everything that we do, we document, meaning we write it down or we enter it into a program real time. And real time means when it happens, not 3 days later, not an hour later, but when it happens. And every single time, no exception. And basically the kind of method that we live by is if it's not written down, it didn't happen. So if you didn't write it down in real time, and everything that you actually do in your documentation, you have someone that is you're checked by. So you perform something, and then your person checks it to make sure that they've witnessed you doing it. So there's a lot of control around how that is all documented. And it's all done in what's called a batch record, which actually in Vicky's experiment, she's gonna show you what a batch record looks like. So every single piece of that process that I talked through, there are probably, I don't know, 30 to 60 batch records that are associated with a single batch. So it's a lot of documentation that goes into it. Those are reviewed by our quality assurance team to make sure everything is following all the rules. And it's audited by the FDA, so at any point we can have the FDA come in and look at something that we're doing. And they have the right, by the way, to tell us that we are not allowed to make it anymore if we're not following those rules. Process controls, so there's strict controls on the process to ensure consistency in manufacturing. So I talked about we monitor things like pH and oxygen levels, all that stuff, really, really tight ranges that we have to operate within. If we don't, and if it goes outside of that range, even by 0.1 of a pH reading, we have what's called a deviation, and we have to investigate it, document it, make sure we understand it. And if we don't understand it, that batch doesn't go through. So it's very stringent and very tightly controlled. Product quality testing, which is Vicky's bread and butter in her team. Do you want to talk a little bit about that, Vicky?
Yeah, so one of the biggest things that we do is, so there's 3 things basically that we're looking for. Making sure it's safe for the patient, making sure that it's going to be effective, and then making sure that everything was done the way it was supposed to be done. So when we're manufacturing a drug, there's a couple things that we have to think about. So the biggest one is what's the impact to the patient. So as we're looking at the different testing that we do, making sure that there's not a breakdown of the protein, making sure that there isn't contaminants in there, making sure that everything was done consistently the same way. So if there's a lab issue, if the test comes back and it's bad, we have to do a really good investigation to make sure that we understand, was there something that fell down in the process? Is there a reason that we can't give that product to a patient? Especially these products, they take very long to manufacture, so we have to make sure that what we're putting into people is the right thing and that we're not going to cause any type of adverse event where we're actually hurting somebody. So a lot of what we do is looking into how does this impact a person and how will this how will this work in the end? So there's a lot that goes into it in terms of making sure, just trying to think. One of the things that we do, so the chromatography steps that we do in there's actually a lab one that we do to make sure that there wasn't anything that happened that causes the protein to fold into maybe not be as effective as it should be. It also can cause harm to the patients if the protein folded over and you have this big monoclonal antibody that's now folded over and not working the right way. It can actually hurt and cause an injection site reaction. So there's a lot of different things that go into it when you're thinking about the quality of the product and how it's going to impact the person.
So how does it make you,
With regards to the FDA approval, once you get approved by the FDA for a specific drug, but then you find maybe in your studies, something happens.
Yep.
Does the FDA continue to monitor you, audit you randomly, periodically?
That's a good question. So there's a couple things that happen. So when we first manufacture a product, we have to do clinical studies. So we file those clinical studies with the FDA. And then when we're ready to take that product commercial, we do what's called a biological licensing application, or a BLA. And that goes to the FDA, and then they come in and they do a pre-approval inspection, or a PAI, and they come in and they check the facility, they check the product, they check all the testing, the manufacturing, everything that goes into it. And then when they say, yes, you're approved. And then what they do is every year we have to do what's called a periodic product review, and we have to send updates to the FDA and show them, here's what's happened in the last year. Here's our deviations, here's the number of batches that we made, and we send that to them to review. Also, on top of that, every 2 years, the FDA will come to our commercial facility and they check for these good manufacturing practices. And they make sure that you have the right documentation, you have the right procedures in place. So not only are we documenting everything, but everything has to be very strictly controlled. So we have basically recipes for everything that we do. And if we don't have a recipe for how we do a test or how we do a manufacturing step, then that's also a problem. So on top of that, there's also things that can happen. So say you do something and you notice that there's a problem with the product, a problem with something that's out on the market, you actually are required by law to notify the FDA and let them know. And that could potentially trigger an on-site inspection where the FDA could come out and look into the issue and making sure if you have to do a product recall, if you have to do anything like that, so that you're not impacting the product or the consumer at the end. So
you can never let up. You need to do that.
Never let up.
With everything you do.
It's constant. It's constant. So the FDA and actually also a lot of the other countries have regulatory agencies. So within the last few years, besides the FDA, we've had folks from the European Union. So we've had people from England. We've had people from Italy. We've also had other countries like Brazil has a regulatory agency, Turkey. So there's a lot of different countries that come out and will actually inspect you on site to make sure that you're doing everything that you're supposed to. Some countries have mutual agreements. So like if England comes out, France might not need to come out, and they'll take what England has done from an inspection perspective. But yes, we never can let up. You can't let down your guard. You always have to make sure that things are being manufactured and done the right way, which is why we have a whole quality assurance team. So not only does my team do the quality control piece, but quality assurance is really responsible for the site and making sure that everything's done the right way. And the FDA comes in, or whatever regulatory agency will come in and make sure that they're inspecting and making sure that what you say is being done is actually being done. Yes.
Do generic drugs get regulated differently?
No, they are regulated the same way. So there's the same thing is done. So they're looking at the active pharmaceutical ingredient as well. But they're also looking at what other controls and changes. They also are required to do clinical studies. They're not quite as robust as what we do for the first time, but they're still very robust.
So how does it make you feel to hear about how strictly controlled it is? As I've been in this industry and learned more and more, it like comforts me to know that.
Yeah.
And now that I have kids of my own and they take medicines and things like that, I feel good about the processes that are in place to kind of keep things very tightly controlled. And the fact that you can never let up is very, very good. So when you hear about companies that don't keep up with that, I don't think you hear about it very often, but when it does, it's actually pretty surprising how it gets to that point. But what's nice about it, I mean, one thing that motivates me each day is I feel like I'm surrounded by, a lot of people, very smart people that want to do the right thing and are very passionate about that and care about the patient. One of the things that we talk about at GSK, and I'm sure a lot of pharma companies do this, is we try and talk about things in terms of the patient rather than anything else that might, be at the surface because you're running the business. It can be very easy to get stuck in the day-to-day. Oh God, another deviation I have to deal with. I have to deal with this, and the pH was out just by this much, and really it's all this work. But keeping that mindset is actually really, really good because it keeps us working to the right goal, which is to keep the patient safe and medication effective.
And especially with the drugs that we manufacture, because a lot of them are people with autoimmune disorders. They're very, they're more susceptible to some of those changes that might happen with the product or things that could potentially impact people. People that are healthier and don't have as many issues, so taking some sort of chemical that might have a side effect might not impact them as much as somebody that is autoimmune compromised, and things like that could impact them severely.
Any questions on that? Yeah.
Where's your lab located?
Our manufacturing facility and our laboratory are in Conshohocken, PA, so right near actually we're really close to the King of Prussia Law.
Which is good.
I have one other business-related question.
Sure.
You had the graphic up there with what Merck's doing, what Novartis is doing. And I remember like a decade ago or so many years ago when the biologics started coming out, and they started coming out like this all across the board. Do you share? How do you share? Do other companies, would Merck purchase something from you that you've developed?
That's a really good question. Yeah.
Or do people just talk?
So there's a couple different avenues that we share. So there's a lot of collaboration between companies. I know GSK has purchased other companies that are some of the startups, so biopharm startups. And sometimes companies will sell off drugs that maybe they don't have the facilities to manufacture, or they'll partner with other companies that they might not have. So if R&D comes up and they have say they have 10 molecules that are potential, and we have space to manufacture 4 of those molecules, but 6 of them turn out to be really good, and we don't have the space for those 2, we'll look to either externalize that manufacturing, or we'll partner with another company to do that manufacturing. So there's a couple different ways that can happen. Also from a science perspective, there's a lot of collaboration that happens, especially in the biopharm industry. There's different forums, like it's BPOG, there's one, that's a biopharmaceutical organization group that does collaboration where we actually will sit down and we'll talk about the next level of manufacturing. So maybe not using a centrifugation process, there's something called an ATF, like a tangential flow, where you can constantly refeed the manufacturing facility and not have to just stop, and you can continue processing a lot longer. So we'll talk about those types of new technologies. Even with the laboratory, there's a lot of different topics we'll talk about. Microbiology tends to be a big topic of discussion because those media-rich bioreactors that feed the cells, they're also really good for feeding bacteria. So if you get something in there, potentially could compete for resources and shut your production bioreactor down. So you have to be really careful about those types of things. So different controls and things that are done are shared across the industry. And there's a lot of collaboration that's done. Obviously, we don't share confidential information or share things that are maybe specific for one type of product, but we will share those types of high-level topics and the next level of technology.
And that's good to know, and that's good for you guys to know too, that science isn't done in a bubble, but that sharing of information where some other company may have what you need to continue the process.
Absolutely.
And that sharing of the minds really helps advance the science forward.
Yeah.
And there's also a lot of examples too of partnering with universities in terms of research, which is also really great to see because, universities are research hubs and it's a great resource to tap into. And there are also other kind of like business venture deals that happen where companies will partner. So for example, the Prolia one that's up there for Amgen, GSK actually distributed that product for a number of years. Novartis and Amgen obviously worked together for the migraine medication that I mentioned. Benlysta was actually an HGS or Human Genome Sciences product, and we ended up buying them.
And I don't see DuPont up there. Didn't DuPont and Merck get together for like a little while and then split off again?
Potentially, maybe.
I'm not sure.
I don't know, actually.
It was like probably a decade ago, DuPont and Merck came together and then DuPont left again.
Yeah, because I don't think they're together now. I'm sure you're right. It's hard to keep track of all of them because a lot of.
But maybe they come together, like you say, to do something, and then once that particular.
Sure.
Because I think it was, they came together to start the HIV thing. So maybe they came together to work on something like that.
Gotcha. Yeah, there's some partnerships.
GSK partner with any university?
We do. I'm trying to remember which ones we partner with. I'm talking about it, Vicki. There's a few in the U.S. and a few in England as well. So GSK is actually a British-based company. So our headquarters are in London. So there are several universities that we partner with, but I probably wouldn't be the best person.
And that's more on the drug discovery side. We do a lot of that where we're kind of on the back end of it, so we don't necessarily see a lot of those partnerships.
But we do also do, like other partner, you mentioned HIV, that's a good one. As a reminder, GSK partners with a company called ViiV Healthcare, which is all HIV products. So what that means is we partner with them on like drug development, manufacturing, and obviously distribution. And that's been a really successful partnership, and some really important products have come out of that for HIV patients. And that's probably been for like 10 years now, I feel like. So yes, there's a lot of cross-sharing that happens. And you mentioned BPOG, that forum. People actually will publish papers together that work at different companies. So I have a few people on my team right now that are publishing papers together on how to, because I work in supply chain, so they're publishing a paper about how to distribute cold chain. So all the stuff that we make is shipped cold, which might sound like a big deal. It's mice packs in it, but it's like a very tightly controlled, very challenging space. So, so it's really cool to see that collaboration across. Definitely science isn't done in a bubble.
That's good to know. It's not like just all siloed, that everybody's working together.
Yeah, especially in biopharma, because I think it's such a young field that people are still learning. So it's for the benefit of everyone if we work together. And like you said, we don't share confidential information because we are separate companies that do compete, but it's good to see that we kind of find that gray area, which is good.
Even vendors will work with a lot of different companies if they have a new type of product that could potentially help. Rapid technologies are a big thing, especially with biopharm. Some of the testing that my lab does can take up to 7 days to get done. So whatever we can do to try to reduce that time and get product out the door to the patient faster is definitely a good opportunity for people to look at. So the vendors, a lot of times will partner with the different companies to try to get a lot of data to help get these new technologies out there.
Any questions?
Good questions.
Yeah. And then, so we're going to talk just a little bit about career pathways in biopharm manufacturing, and then maybe Vicki, we can talk a little bit about our career pathways, just for you guys to kind of hear a little bit about that. So some of the degrees that you could have and kind of go into biopharm manufacturing, if it's interesting to you, is biotechnology, chemical engineering, bioengineering, mechanical engineering, biochemistry, micro, and biology. There's the research and development aspect, which we haven't talked about too much because we've talked mostly about the manufacturing side of things. But there's a lot there in terms of drug discovery, manufacturing process development, so true like laboratory bench work, running experiments, actually discovering products and making them, developing manufacturing processes so that they can be made and given to patients. On the manufacturing side, some of the departments within our site where we work is production, so the people that are actually on the floor manufacturing, quality control, which is Vicki's area, technical, so there are people that do kind of the more technical side. They do a little bit of bench work, process design, or run experiments to help improve our already existing manufacturing process and help us troubleshoot any issues that come up. We have a lot of engineers. We need a lot of engineers to run something like this because you have a lot of equipment, and those are the folks that do our very specific equipment design, process troubleshooting, so very important component. And then of course quality assurance, Vicki talked about, so they're the ones that are kind of governing us every day, making sure we're doing the right things. And then, yeah, I'm just going to talk maybe a little bit about our career pathways, just to give you guys an idea. So I started at Penn State after I graduated from Weiser, and I majored in biotechnology, which at the time was a pretty new curriculum, and we had a pretty small class. But it focused on this kind of large-scale manufacturing in the outer years of when I was in college, and I was really interested in it, and I ended up getting a job at GSK right out of college. And I started actually as a manufacturing associate in production, working on the floor. So I was when you work on the floor, and we'll talk a little bit about this when we do the experiment, but you gown up in scrubs and all kinds of stuff to cover up so you're working in a sterile environment. And that's what I did for my first couple years out of college, and it was great because I learned so much about manufacturing process. And then I moved into a specialist role where I was writing deviations and doing things like we've talked about, and I've been there for 13 years. I've actually moved into supply chain, which is, if you would have asked me when I was graduating high school if I would be doing something like that, I would have thought you were crazy because I didn't even know what supply chain was. But it was just kind of where my career path took me. GSK is, I'm lucky because it's a big company that has a lot of different career avenues that you can take. And I've had really great support in kind of moving and developing into different parts of the career. So I got a little bit of a taste of different parts of the manufacturing side of things. And then when I was at GSK, they actually paid for me to go back to grad school. So I went to University of Penn and got my master's in biotechnology. A couple years out of college, I did it part-time, and it took me, I think, 4 years, and I just did, a class at a time. So it was great because it was close by to where I was working, and has been really helpful in kind of understanding the technical aspects of it more. So that's kind of me in a nutshell.
So I went to Albright College, and I have my degree in biology. From there, I started working in a contract lab. I've worked in different types of laboratory environments for the last 20 years. 10 years I've been at GSK, very focused on microbiology in the beginning. So after I started working, I actually had GSK paid for me to get my master's in microbiology, which is a very good opportunity if you guys are thinking about furthering your careers after college, potentially working at a company and seeing if you can get a company to help you further your education wherever area that you're interested in. So from there, I've done a lot of different things. So right now, my laboratory not only does microbiology, but we also do biochemistry. We look at raw materials coming in, and we also look at the utilities and the environment for the entire facility. So when you think about the manufacturing facility, we use a ton of water. So that water isn't just the drinking water that you get out of there. There's a lot of processing that has to go through, and we actually look at that like the raw material. So a lot of testing has to be done to make sure that that water is okay to go into people, and a lot of stuff that has to come into the process has to get tested before we even go through the production facility. And then we're checking in process, making sure that those purification steps are removing impurities and looking at those different things. So for me, it's been a very interesting path. So I started very targeted and kind of opened up my career a little bit more to learn about the different aspects. I also have learned over the years how the quality control laboratory impacts production and how the different steps can influence the testing that we do. So it's really important to kind of understand that holistic piece of it and be able to help aid in investigation. So if we do see a bad laboratory test, we can help production figure out what happened and making sure that they're able to adjust and be able to release product to the patient. So. Any questions?
That's it. Okay. Thanks, guys.
So I would sit in the back the whole time, just to take a couple things out of it and how we can apply to SRI and Explore Coderlizer. So one of the exciting things about being at Coderlizer is we can do a lot of the stuff that they were talking about. So we do cell culture. We do some microbiology work, like Matai, we were just growing E. coli the other day. You guys work with fruit flies on occasion, right? We work with C. elegans. We can grow yeast, but we can also grow, we usually do Dicty cells, so protocells, insect cells. Right now we have horse and mouse cells that we grow in culture. We don't do any human cells, but so from that part of it, if you were interested in any piece, we can actually do that here. We do a lot of that purification stuff. We're obviously not producing pharmaceutical drugs, but if you're interested in any one of those pieces, you can start to experience that now. Very few other, if any, middle school and high school students have the opportunity to do those steps. So if you're interested, let me know, and we can get you hooked up with the right people to do that. The other thing that I think is really cool, I see a lot talking to high school students, is like if you're sitting here thinking like, I want to be an engineer. I remember thinking when I was in high school, like, okay, if I want to be an engineer, I go to college for engineering, and then I work at an engineering firm. Or if I want to, like, we like to think, or we think that the career pathways are very straight. And hopefully you heard, like, they're not. Like, if you're interested in science right now, but you're also kind of interested in business, you can combine those two. If you're interested in engineering, you might be able to end up working for a company like this. So don't think that if you have 2 or 3 different career options floating around in your head, that they can't be combined into one, which I think is really cool. So.
And I think another thing just to add on to that too is when you think about your career path and you think about, if I'm going to work in a lab, I'm going to come into the lab and do the same thing every day, day in and day out, I'm just going to run this test. It really isn't like that. There's different opportunities, not only to develop what assays that you're doing or making changes, and you can learn so many new technologies, you can bring in new technologies, there's so many different things that you can do. And I think if you ask the folks in my laboratory, do you run the same thing every day? Is it boring day in and day out? It really isn't. There's so much opportunity to learn and to develop and grow. Even people that don't want to become leaders in the laboratory, you can learn different technologies and continue to be excited and learn new things when you come to work. And with Erica, I think Erica's a really great example in doing supply chain now. She used her science degree to really propel her into something that really impacts the overall, how the drugs are sent out to the patient, so.
And I'm glad you said that too about the, what you were thinking in high school, because I wish I had that mindset when I was in high school, because it stressed me out to think like I have to pick this one path and there's so many to choose from, and it's just such a big decision to make at that age. And I wish I hadn't maybe been so focused on that, because I would have been a little more relaxed, I think, because I was so sure that I wanted to work in a laboratory in like drug discovery and do that. And then what's funny is when I was in college, I actually got like a part-time job in the lab doing that, like working in like research. And I didn't like it. I actually found out that I didn't like it, and it felt like a failure almost, because I was like, well, that's what I was supposed to do. That's what I wanted to do. It just turned out that, sometimes the experiences that you get along the way will be so valuable, even the ones where you figure out what you don't like, because then it'll actually drive you to the thing you are passionate about. So that's been one really big learning for me, is like, okay, you'll lead yourself somewhere that's going to be good, and especially when you work for, like our company has been great about that sort of development. A lot of companies are, and working for one that when you get to that point in your career that actually cares about your professional development is really important. It's important for you to be selfish in that way and want to get the most you can out of it. It might not feel like that when you're like low on the totem pole, but keep that mindset, because the companies kind of owe that to you, and it's best for them to invest in you and figuring out what you want to do when you get there. Yeah, so don't feel too much pressure.
I think it's great you guys have an opportunity to try different things here. You guys really are in a unique environment, because a lot of schools offer this type of exposure to these different types of technologies. So try to get in there, do different things, and see what you like. You might find that, I really don't like doing this, or I really do love doing this. And you don't have to go down that one career path. You definitely can see what you like and see how you can expand on it and use your strengths and really grow in a career for yourself. So.